COVID-19 has the world in lockdown, and the core reason is because we do not know who carries the virus and who does. To make matters worse, some carriers of COVID-19 do not show any symptoms even though they can still spread the disease. Now, here's the real kicker. We can actually test if someone carries the virus. So, why don't we test everyone? What can we potentially do to test everyone? Currently, when someone is suspected of COVID-19, medical personnel take a sample swab from the patient and then send the swab to a specialized testing laboratory. In this testing lab, which is rated for biosafety level 3, a technique called RT-PCR is used to detect any potential viral RNA that is produced in cells infected with the virus that causes COVID-19, known as SARS-CoV-2. In case you don't know, RNA is a lot like DNA, but with a few small structural differences. So far, so good. Except that it is expensive. Results can potentially take days, and we do not have the capacity to test everyone. Clinical tests for SARS-CoV-2 are costly and limited because of the need for medical professionals to administer the swabs and the safety precautions needed for a biosafety level 3 laboratory. All of this translates to significant expenses in terms of human resources, personal protective equipment, transport, and highly specialized facilities and equipment. And thus, you are stuck at home, suffering from cabin fever, and for some of us, even jobless overnight. But it may not have to be this way forever. Scientists from South Korea at the Korean Advanced Institute of Science and Technology in Daejeon and other collaborating institutes have proposed a low-cost and highly scalable detection method for SARS-CoV-2 that can potentially test significantly more asymptomatic members of the public. To overcome the problem of requiring medical professionals to administer the swab, the person being tested will self-administer their own swab in a ventilated fume hood under precise instructions. While next to the fume hood, the person being tested will use a solvent to extract some of the potential virus from the swab into a test tube, and then immediately discard the swab in a biohazard waste bin. Then, this person can leave the room. The reliance on self-collection and the use of fume hoods is crucial in offsetting the need for medical workers and the associated PPE that comes with protecting them. This combination helps reduce cost and the time of transport. Now, on to reducing the need for highly specialized biosafety level 3 laboratories. Biosafety level 3 is required for COVID-19 lab work because SARS-CoV-2 can spread via aerosols suspended in the air. Biosafety level 3 labs are much more stringent in requirements. For example, they require solid front protective clothing for personnel working in the lab. And hence, there is a significantly smaller number of biosafety level 3 as opposed to biosafety level 2 labs available. Back to the process, lab staff will add a chemical mixture known as trizol, which inactivates the virus within the test tube sample. And surprise, trizol is the same chemical solution used to extract the viral RNA before performing RT-PCR. By cleverly using trizol, which extracts RNA while inactivating the virus, and by providing the fume hoods needed for patients to self-administer the swabs, the need for a biosafety level 3 lab for the subsequent testing processes is eliminated. Potentially, every lab that has a fume hood and a PCR machine can now be converted into a COVID-19 testing facility, or in fact, any other type of pandemic testing facility. Pretty cool, huh? This proposed method can help potentially reduce the cost of testing for SARS-CoV-2 that is responsible for COVID-19 to around $15 per test and a turnaround time of 4 hours. And best of all, it eliminates the need for high-level biosafety labs, potentially allowing for other lower-level labs to be converted into COVID-19 testing facilities so that we can all get tested have peace of mind, and put this lockdown behind us. The paper we covered in this video is titled Development of a Laboratory-Safe and Low-Cost Detection Protocol for SARS-CoV-2 of the Coronavirus Disease, 2019 COVID-19, published in the journal Experimental Neurobiology. Details of the paper can be found in the description below, and we encourage you to read the paper. Obviously, we can't cover the whole publication in 5 minutes, but we hope you're intrigued enough to find out more.